Okay, do we need the lights on in here or is it okay? It's okay? Right. So, hello everyone again. Um, so this morning what we're going to do is finish up with the molecular aspects of virus replication, I guess. So I'll just complete the strategies for genome replication and expression, and then we'll go on to mechanisms of assembly and release of different types of virus particles. So to finish up on strategies of genome replication and expression, the last one that we need to consider, or that last type of case, are retroviruses. So these are viruses that have a plus strand RNA genome in the virus particle, but in the infected cell, they replicate via a double-stranded DNA intermediate. So retroviruses is quite a diverse family with spumoviruses and orthoretroviruses. And among the orthoretroviruses, we've got like 1, 2, 3, 4, 5 genera plus lentiviruses, so 6 genera. All of them have got enveloped icosahedral capsids, and in lentiviruses, you've got this very specific conical shape of the capsid. Now, one thing kind of interesting about retroviruses is that the capsid contains 2 copies of the genome. So in a sense, these viruses are kind of like diploid. Okay. So if you ever get if one cell gets infected with 2 different strains of HIV-1, for example, then inside the capsid you can have one genome copy from strain A and another genome copy from strain B. And during reverse transcription, they can recombine, in fact. So there's an interesting mechanism to generate genetic diversity that retroviruses have, not necessarily true of other types of viruses. So recombination occurs during the during each round of reverse transcription, in fact. Okay, so in humans, I mean, we don't have very many retroviral infections. So up until the beginning of the 1980s, there was only HTLV1, which was known, human T-cell lymphotropic virus. And in the beginning of the 1980s, so with Françoise Barry-Senoussi and Luc Montagnier discovered HIV-1 at the Pasteur Institute. And out of these 2, there's evidence that HTLV1 has been infecting humans for at least a few hundred years. But HIV-1 is a recent introduction. So this is a zoonotic event. So it's a simian virus that was transferred to humans with multiple transmission events. So there are HIV-1 and HIV-2. There are 2 distinct species of human immunodeficiency virus. And within HIV-1, the main clades are all derived from different transmission events from chimpanzees. And this was within the last 120 years or so. Okay, so up until like the last century, the human population probably only was infected by 1 retrovirus. Okay, so one other thing I wanted to say about this is, but of course, in our like evolutionary history, there have been a lot of retroviral infections because a large part of our genome is constituted of endogenous retroviral sequences. So at some point in our past, perhaps millions, tens of millions of years ago, retroviruses have infected our ancestors, somehow got access to the germline cells, and being transmitted across generations as a provirus integrated form. So most of these endogenous retroviral elements are inactivated, but some of them seem to activate their transcription during during early development. Okay, so what's the kind of general idea of retroviral replication and gene expression? So the genome is plus strand RNA, and it's got to copy itself into a double-stranded DNA provirus. So this depends on an RNA-dependent DNA polymerase, a reverse transcriptase. And because this occurs directly after infection, once again, the virus has got to carry the reverse transcriptase inside the particle, so that directly after infection, the genome gets copied into double-stranded DNA. Then afterwards, this double-stranded DNA will be retranscribed into RNA because there is a promoter region in the proviral DNA that can direct the host RNA polymerase II to transcribe messenger RNAs from the proviral genome. Now, one of the issues is, how do you regenerate a complete genome at every cycle? Because, if you imagine you've got the promoter region here at the 5' end of this provirus, then the transcription will start a little bit further along. And normally, you won't be able to copy the promoter region. So it's not at all obvious how retroviruses can manage to regenerate a complete genome if they do this. And so that's genome replication. For genome expression, that's kind of easier because all of the messenger RNAs use the host RNA polymerase to transcribe their messengers. The virus proviral genome is in the nucleus, so all of the splicing and the mRNA maturation machinery is available. One of the problems retroviruses have is, how do you express multiple proteins, multiple mRNAs from a single promoter? Okay, because we just basically got one promoter region here to drive the expression of all the virus genes. Okay, so for replication, if you look at the structure of the genomic RNA in a retrovirus particle and the integrated proviral DNA, you'll notice that there are some differences. So the coding part is in the middle. All of the open reading frames that code for proteins, they're in this red region here. And there are repeated regions at the 5' and the 3' terminal end of the RNA genome. Next to these are two untranslated regions, untranslated 5', that's U5, and untranslated 3', that's U3. Now, what happens during reverse transcription is there's a kind of there's a really neat mechanism by which the virus can copy the U3 region to the other end of the proviral DNA, and also the U5 region gets copied at the end of, the other end of the genome. So in the double-stranded DNA provirus, you don't have this RU5 and then U3R. You have the same sequence, the same long terminal repeat sequence, LTR, at each end. And then this is what gets integrated into the host genome. Okay, so this is the trick, how to regenerate a complete genome at each cycle, because the promoter region is in this U3 segment here. This is where the host cell transcription factors bind. So the transcription factors bind here, and transcription occurs from the beginning of the R segment. It goes all the way through here, and then you get a poly A signal at the end of R over here. So that means that this, when the provirus is transcribed by the host cell RNA polymerase, then you regenerate a full genome with R, U5 at the 5' end and U3R at the 3' end. And it's because there's a mechanism during reverse transcription that allows the virus to recopy this region over here and recopy this region, the U5 region at the other end. Okay. So I advise everybody to go check out this video from Vincent Racaniello where he explains it really well, and you can see how the LTRs get generated. So that's the genome replication. How about gene expression? So here the example is the HIV-1 genome. Now, all retroviruses have got the same genome expression with 3 large open reading frames coding for GAG, POL, and ENV polyproteins. So ENV, that's for the envelope glycoproteins. POL, that's for polymerase, so it codes for the reverse transcriptase and the other enzymes involved in virus replication, which are the integrase and a virus protease. GAG, it's a rather obscure acronym, but it means group antigens. And these group antigens are the capsid antigens. So the matrix, the capsid, and the nucleocapsid that form up the inner part of the virus particle. Okay, so all retroviruses have got GAG, POL, and ENV. And then different retroviruses have different accessory proteins. So in HIV-1, you've got VIF, TAT, VPU, NEF, all of these things. So these will be different for different retroviruses. Okay, HIV-1's got these. Not all retroviruses have this collection of accessory proteins. So how do these different proteins get expressed? Well, there's a full-length RNA that starts to be expressed here. So you've got the U3, R, U5 regions. So the mRNA starts from the R region, gets capped, and the full-length transcript goes all the way through to here. And that will code for the GAG and the GAG-POL polyproteins. Okay, because this is the first start codon here. So the proteins that will be expressed from the unspliced RNA are these ones. And then all the other proteins will be expressed from spliced messenger RNA transcripts. And alternative splicing allows the virus to express several different messenger RNAs from the first primary transcript. So the envelope glycoproteins, they're expressed from a mono single spliced messenger RNA, and there are multiple spliced messenger RNAs that give you different proteins. Okay, so that's 2 ways to get different proteins from the same transcript. And then for the unspliced messenger RNA, how do you get multiple proteins from here? Well, you have the GAG and the POL reading frames, that are, the GAG and the POL coding regions, which are in different reading frames. Okay, so that's what is indicated by this diagram here. So each time you move up a level, these are the different reading frames on the RNA molecule. So if you start transcribing, if you have this messenger RNA and the ribosome starts translating the GAG polyprotein, normally what will happen is that they will come up to a stop signal, stop codon here, and that will be the end of the protein translation. However, what can happen in about 1 in 20 times, one time in 20, is that the ribosome will kind of like be slightly delayed by secondary RNA structure here, and it will pull back one nucleotide. So the reading frame will change to +1 or -1. Oh, I can't. Okay, but it skips reading frame, okay? And that's how it can read through this stop codon, because it's not stop codon anymore. It's split into two different codons coding for 2 amino acids. So 19 times out of 20, the reading frame is maintained and you get a stop codon. 1 time out of 20, the reading frame changes and the ribosome will read through to translate this GAG-POL polyprotein. So you get these two polyproteins translated, and within the POL reading frame is the virus protease, which will cleave both GAG and GAG-POL into the individual protein subunits. Okay. So you've got two things going on here. There's using two different reading frames to get a short and a long polyprotein, and then the protease cleaving them. So this is kind of makes sense, right? Because it's just like how we ended with the negative strand RNA viruses, the mononegaviral. For an infectious virus, you need more copies of the capsid proteins than you need of the polymerase. Okay, so it makes sense to express 20 times more capsid proteins than you have polymerase. So that's why it's like this, okay, and is not the other way around. Okay, so that's about it. So let's just see if you've picked up on this. So I just think about these and we'll have a show of hands. Are we ready? Yeah, so retroviruses can express different genes from different promoters. Who says yes? Nobody, you're right, okay. One promoter in the LTR. They express a polyprotein cleaved by a viral protease. Who says yes? Yeah, that's right. GAG and GAG-POL, it's exactly that. They express different mRNAs by alternative splicing. Who says yes? Yeah, that's right. Single spliced or multiple spliced at the end when the accessory proteins, GAG, GAG-POL, are expressed from the unspliced genomic RNA. You get different proteins from a single mRNA by translating through a stop codon. Who says yes? Yeah, that's right. You can express different proteins from a single mRNA using an IRES, internal ribosome entry site. Who says yes? You're right, doesn't happen. Okay, so retroviruses, they're kind of a mix of what we saw already for DNA viruses and plus-strand RNA viruses, because using alternative splicing, the DNA viruses use this one, polyprotein cleavage by a protease, we saw that in the picornaviruses and in the flaviviruses. And if you kind of look at it, look at the genome organization, this part looks a little bit like a plus-strand RNA virus genome, because you've got the structural proteins in the 5' part and the enzymes involved in replication in the 3' region. So the way it's organized is pretty much the same as like a picornavirus, and the whole thing is in a big polyprotein. So, well, I don't know if this is an actual, strongly conserved evolutionary relationship, but it's interesting, I think. Okay, so summary for all of this. So DNA viruses, one thing to remember, there's a big clear relationship between the size of the virus genome and how much the virus depends on the host cell for DNA replication and gene expression. So the more independent the DNA virus is, the bigger its genome has to be. The small DNA viruses always need to use the host cell mechanisms for DNA replication. And the other thing is early, late virus gene expression through kind of a cascade of control mechanisms. Plus strand RNA viruses, RS is a big thing, can be cap dependent in different viruses. And then the common theme is polyprotein and viral protease, cleaving it up into individual proteins. So for the negative strand RNA viruses, the only one that was really presented was orthomixoviruses. And one of the things that is important is there's a difference between the plus strand copy of the genome, which is not capped, doesn't have poly A, is just used to regenerate the genomic negative strand RNA, and messenger RNA. So to get the messenger RNA, influenza virus steals the 5' cap from cellular messenger RNAs. Retroviruses, the main kind of cycle between RNA, double-stranded DNA, and RNA again. And the combination of mechanisms for expression of different genes, there was a kind of mix of what we saw in plus-strand RNA viruses and double-stranded DNA viruses. So not only, you know, the genome of retroviruses is a kind of weird mix between a plus-strand RNA virus and a double-stranded DNA virus, but also the mechanisms used to express different genes are a mix of those two things, or the strategies we found in those two types of viruses. Okay, so this is kind of the last of the mechanistic lectures, and it's at the end stage of the virus replication cycle. You've expressed viral proteins, you've replicated the genome, you've got to put them together into an infectious particle, and these particles have got to be released from the infected cell. So we're going to look at sites of virus production, how capsids are formed, packaging signals. Google Translate, you betray me again. Packaging signal, packaging sequence. Sequence, don't press it. Yeah, packaging sequences. Okay, let's just correct that. So we got packaging signals, packaging sequences, release of non-envelope particles, and budding of envelope particles. So production sites, one of the things that is a common theme for all viruses is that, okay, we'll talk about this later. So DNA viruses, so most of them are in the nucleus, right? They replicate in the nucleus. The genome will be replicated in the nucleus. Now, as I think basic, yeah, let me just draw this out. So what are the two possibilities? Okay, okay, I was given the answer here. So all DNA viruses that replicate in the nucleus, they also assemble their particles in the nucleus. And that means that all the virus proteins have got to be imported into the nucleus, all the virus capsid proteins. So they all have a nuclear localization sequence in the capsid protein. Now, if you kind of think about it, it's not necessarily the case, right? Because you could think of a cell here, right? And we're replicating the virus DNA. And you want to make the capsid proteins, obviously, they get translated in the cytoplasm. So you've kind of got two options. Either the protein can come inside or the DNA can go outside the nucleus. And you can assemble the particles here. No DNA virus exports its DNA for capsid assembly. If I were to guess why this is the case, it's probably because a lot of the antiviral innate immune mechanisms are based on detecting the wrong type of nucleic acid in the wrong place. So you know, if you've got any double-stranded DNA in the cytoplasm, the cell is not going to like this at all, okay? So it's kind of better for viruses to import the proteins and form the capsids there into the compartment where it's normal to have DNA. Okay? So all DNA viruses do this, except for those that are the large DNA viruses that replicate in the cytoplasm. However, as I just kind of suggested here, having double-stranded DNA directly in contact with the cytoplasm is going to alert the cell that it's been infected by a virus. So these large DNA viruses replicating in the cytoplasm, they kind of sequestrate their DNA replication in a kind of mini nucleus that the virus produces inside the infected cell. So this is an electron micrograph of cells infected with a pox virus 2 hours post-infection. You see this new kind of structure arising in the cell, and this is where the virus is replicating its DNA. It's keeping it separate from the cytosol. Okay? So you have these kind of like centers of DNA replication to prevent the cell from realizing I've been infected. What about RNA viruses? So they have the same kind of thing. So up to now, or in the kind of cartoon version of virus replication, I've got a big box, that's a cell, and we've got a load of copies of virus genome everywhere. But in reality, viruses will concentrate their RNA or DNA replication in a specific site. And this is kind of like a virus production factory. So enveloped plus-stranded RNA viruses have their viral production factories that are associated with the endoplasmic reticulum. And it's also true for picornaviruses. They're closely related to the endoplasmic reticulum. So if you look at an electron micrograph of cells infected with dengue virus, you can see that the endoplasmic reticulum forms these unusual invaginations. You can see these kind of like little round balls here that appear to be empty. And if you kind of zoom up on them, you can see that this kind of ball is continuous with the cytosol. Okay, so this is the cytosol here. This is the lumen of the endoplasmic reticulum. And you've got a kind of like sphere that's been kind of created here, but the neck is open. So this is continuous with the cytoplasm. And so you have all these kind of unusual structures associated with the endoplasmic reticulum. And then nearby, you have regions where you've got, which are packed full of newly formed virus particles, new virions here. And if you get a 3D view of it, it's kind of like this, kind of cool. You have these regions packed with viruses. So what appears to be happening here is that inside these kind of like empty balls here, this is where the virus RNA replication is occurring. And once again, the idea is you don't want the cell to realize you're replicating a load of virus RNA here. You kind of hide it away in a specialized compartment. And then the RNA, once it's been replicated, it will be incorporated into capsids that are being formed by budding at the endoplasmic reticulum membrane. So the capsid, matrix, and envelope proteins of flaviviruses, they're at the 5' end of the mRNA, so at the N-terminal part of the polyprotein, and they are always associated with the endoplasmic reticulum membrane. So as the virus particle forms here, the RNA is transferred from this compartment into the new virus capsid, which then goes out through the secretory pathway and is released. Okay, so that's how plus strand, well, one example of a plus strand virus does this. Negative strand RNA viruses, once again, you get the same kind of thing, especially for the mono-nega viral. These monocatenary negative strand RNA viruses like rabies virus. Once again, the RNA replication isn't distributed homogeneously throughout the cytoplasm. There are regions inside the infected cell where all the virus RNA replication is concentrated. And this is what causes the characteristic inclusions that you can see in infected cells. So this is a kind of a biopsy or a necropsy sample of a person or perhaps an animal that was infected with rabies. And you can see these neurons which have these unusual inclusions here called Negri bodies. They are characteristic of infection by rabies virus. And if you look by electron microscopy, you can see that these are these regions of a kind of amorphous ribonuclear protein. And then you've got budding out from here, you have the formation of new virus particles coming out here. Okay, so rabies virus has got a very typical bullet-shaped nucleocapsid, kind of like this. So, negative strand RNA viruses also will have these kind of like virus production factories in the cytoplasm. So the exceptions are the orthomyxoviridae, where the virus has got nuclear replication. So the nucleocapsids are formed in the nucleus. Okay, so that's the basic idea, but how do you form the nucleocapsid? Because it's a structure made out of proteins. And what comes off the ribosome isn't a fully formed capsid, it's a capsid protein. So somehow these capsid proteins have got to be put together, pulled together into a particle. Now, a lot of viruses have, or virus capsid proteins rather, have self-assembly properties. So if you express them in a recombinant format, then just expressing the capsid protein or capsid proteins allows them to assemble and give you a virus particle. And that's kind of useful. So papillomaviruses, this works. Polyomaviruses, it works like this. Picornaviruses, kind of. And the advantage of this, for biotechnology anyway, is that you can form virus-like particles, VLPs, just by expressing the capsid protein. And that's why the HPV vaccine works, because you can take the major capsid protein L1, express it as a recombinant protein in yeast, and it forms particles that look exactly like the real virus. Okay, so that's useful in biotechnology. Now, some capsids don't associate spontaneously. So herpes viruses, they construct their capsids in two steps. First, around a scaffold, and then the scaffold protein is degraded to leave an empty capsid, which is then filled. And then other viruses will add capsid subunits around the nucleic acid. Okay, so it's the interaction between the virus genome and the capsid proteins which will start the process of capsid formation. So that's true for viruses with helical capsids, like as we'll see, tobacco mosaic virus, plant virus, and all the negative strand RNA viruses. Also true for retroviruses, to a certain extent. Okay, so self-assembly. Well, am I missing a slide here? Ooh. Yeah, actually, probably it would make sense to do this in a different order. It doesn't matter, okay. Because I kind of, I should do this in a different order, right? Okay, so the simplest case is that the capsid proteins just stick together already. So that's true for polyomaviruses and papillomaviruses. There are two types of capsid proteins, the major capsid protein VP1 and then VP2 or VP3 in polyomaviruses. So VP1 will form a pentamer, and each pentamer is associated with one copy of VP2 or VP3. So this occurs in the cytoplasm. You get pentamers formed. Then they're imported into the nucleus because they have nuclear localization sequences or signals. And inside the nucleus, they form into a capsid. So caliciviruses, like human noroviruses, do the same kind of thing. The plus-strand RNA viruses, the capsid protein is formed as a dimer, and then 90 dimers assemble just like on their own into a capsid. So it's an entirely autonomous system. Once again, if you take this capsid protein, you express it in a different system, it will form empty particles. So slightly more complex is the picornaviruses. So if you remember, picornaviruses express all their proteins firstly as a polyprotein. And then the cleavage of this polyprotein follows an ordered step. So you get like, there's your polyprotein. And the first thing that happens is that these three blocks will be cleaved. And this one at the N-terminal part of the polyprotein codes for the capsid proteins. So as the whole thing comes off the ribosome, they're initially all connected together, but the first cleavage will give you this kind of association between VP4, VP2, VP3, and VP1. So they're all associated together as soon as they get synthesized. And that means they don't have to like find each other in the cytoplasm. They're all together immediately, right? So they all cluster together and form what's called a 5s particle. So this is to do with the sedimentation velocity during ultracentrifugation. That's what this number comes from, okay? So this 5s particle is just an association of these four capsid proteins. And then they get cleaved. So firstly, VP3 and VP1 are cleaved off, and that leaves you this situation. But they all stay together, right? Because they were all stuck together right at the beginning. So this kind of slightly modified form of the capsid protein tetramer can associate together. So you get 5 copies of this to form a kind of pentamer. And that's what's called the 14s particle. Now, the 14s particle can associate together into an immature virus capsid. So this is the 75s particle. But this is a reversible reaction. So you've got to try and imagine, you've got 2 little arrows here, one going this way, one going that way. It's an equilibrium. And finally, what's going to happen is at the end, so this can incorporate virus RNA, but the final step is the last cleavage, and this is what's going to lock. Yeah, I don't know, this one seems to have no RNA in it because it's too light, right? So here, the sedimentation coefficient is telling you something about the density of the particle, right? So this one has got the RNA in it. It's the same size, it's denser though. So the maturation step is catalyzed by incorporation of the RNA and the final proteolytic cleavage here. Okay, so it's kind of autonomous, but the proteins on their own aren't sufficient. Okay, so how about herpes viruses? So this is an example of construction around a scaffold. So this is all happening inside the nucleus. And the first thing that is going to happen is that the capsid formation is going to start with this portal complex. So if you remember, in a herpes virus capsid, you've got an icosahedron, but one of the vertices is going to be constituted of this portal. This is where the DNA comes in and goes out. So the capsid formation starts with the portal, and it's going to associate with the major capsid protein associated with the scaffold and a protease. And in fact, the interactions that allow new subunits to be added are really catalyzed by this scaffold protein. So the scaffold protein, if you don't have the scaffold protein, the whole thing doesn't assemble. Okay, so the interactions between adjacent subunits that are getting added, they depend on the scaffold protein. So this will give you, firstly, what's called a procapsid. It's got protein, and it's got on the outer layer, and it's also got protein inside. Now the problem here is there's not enough space for the genome in there. Okay, so you've got to get rid of this scaffold protein, and that's the job of the protease. It's also incorporated. So the protease cleaves off all of this, and the scaffold protein can pass through the portal. Okay, it's like a pore, and the scaffold protein gets out through it. So the protease is removed, the scaffold protein is removed. Now, if that works correctly, all of the scaffold protein is removed, and the portal will associate with this terminase enzyme. And the terminase is the enzyme that will pack the viral DNA into the capsid. Once again, this is kind of reversible step. The DNA can go in, the DNA can go out, but the process is completed when the terminase complex detaches. So once the terminase complex detaches, this portal is closed, so the DNA can't come out anymore. So this is a complicated process. You've got, first you form a capsid that's got protein on the inside, has to be cleaved to give you something that's empty. And then once you've got an empty capsid, you can put the DNA into it with the terminase complex. So, oh, there we go. I forgot I had text on this. Yeah. So this general mechanism is the same thing with T4, bacteriophage, lambdaphage, all of these like tailed phages. They've got pretty much the same kind of mechanism to produce their capsids and fill them up. So this is one of the reasons why, you know, they're grouped together in the big class of the duplo DNA viria. Okay, so imagine we want to produce VLPs of a picorna virus. How? Yeah.
Sorry, I just had a question. So A capsid and B capsid are not functional?
No, they're not. So A is completely empty, no DNA, and B is partially empty. The protease hasn't cleared out all the scaffold. And so these are also kind of reversible steps here, because actually I think they're reversible. So the terminase complex can attach here and put the DNA back in, I think. And
doesn't that mean that the terminase can also reattach to C capsid and make the DNA go out?
Yeah, maybe. So maybe this is just a dead end side. I would actually have to read this paper in more detail to find that out. So it's possible. The way the arrows look, it looks like this is the main pathway, and these are like side products that don't lead anywhere. Yeah, that would make more sense. Okay, so imagine we're want to make some VLPs from a picornavirus. What are you going to need to express here? Everybody okay with this? Okay. Give us the polyprotein. That will work. Who says yes? No, it won't work. It will get stuck at the 5S particle stage. Polyprotein and the 3C protease. Will that work? One person says yes. Get all the proteins individually. Will that work? Nah. Okay, they won't really find each other. You won't even be able to get the first step going, or it won't be very efficient. And then you put polyprotein, 3C protease, and viral RNA. Who says that? Nobody. Okay, so we only had one person expressing an opinion. Okay, so you can figure out which ones won't work. Okay, this one won't work because you need the protease step to cleave them in the process. This one won't work, although I put it in here because I wondered maybe this would work, but probably it will not be efficient because you would have the different proteins being synthesized from different messenger RNAs in your artificial system. So they are totally separated somewhere in the cytosol. And the first step, having them associated together, will probably not work very well here. Okay, so being expressed as a polyprotein to begin with is probably important. Although I'm not sure anyone really tried this. So that leaves B and D. Do you really need the RNA or not? So you need the RNA to get an infectious particle because otherwise you've got no genome in there. But if you just got the protease, you can at least get to this stage. So you can get to a kind of particle that hopefully is quite stable. Well, might not be fully stable until you get the last maturation step. So in fact, people who do this, this is the way it works. You have one plasmid codes for the polyprotein, one plasmid for the protease, and it's enough. But you can't just use the capsid proteins on their own. They can't associate spontaneously. Okay, so that's how capsids are formed. Now another issue for viruses is you have to put in the virus genome, specifically the virus genome, or not just any old bit of DNA or RNA that is lying around in the cell. So you've got to have some mechanism to be specific about this. So there are several viruses that use packaging sequences, which are called the Psi sequence. But there are several viruses that don't have a packaging sequence, although people have looked for one. So how do they get their genome specifically into the capsid? And then influenza virus has got another problem because it's got its genome distributed on 8 different molecules of RNA. So that's also going to be a bit of an issue because you need to have one copy of each. So we'll look at this. So the first way to get specific packaging of the virus genome is to have a sequence somewhere in the genomic DNA or RNA that is recognized by the capsid proteins. So the main model for this, or the first model, was the way tobacco mosaic virus packaged its genomic RNA. So the packaging sequence, the Psi sequence in tobacco mosaic virus, forms this hairpin loop structure, and this secondary structure binds specifically to the capsid discs. And then once the RNA interacts with the disc, then it will kind of dislocate one of these 2 rings and that frees up a face of a protein monomer, so other ones can add on here, and that's what seeds and what initiates the capsid formation. So specific interaction between the capsid proteins and secondary structure in the virus genome. Now there's something similar in retroviruses, because retroviruses have a packaging sequence somewhere in the LTR. Or no, that's not true. Somewhere in the genomic mRNA. Yeah, it's kind of here. So the example here is for HIV-1. For different retroviruses, it can have a different location. So there is secondary structure in the RNA, which is going to be recognized by the viral nucleocapsid. And for HIV, what is clever is that the packaging signal is, or rather covers, the 5' donor splice site. So if you remember, the genomic RNA is the unspliced messenger RNA. So the genomic RNA, it's got the whole genome coding for all the different proteins. The spliced RNAs, they just code for a little bit. But of course, upstream of here, this is going to be shared between the spliced and the unspliced mRNAs. So you don't want your packaging signal here, because you'll be able to put in a whole bunch of spliced mRNAs that don't contain the whole genome. So here you've got the 5' spliced donor site, which is here in the middle of the packaging signal. That allows the virus to specifically package the full-length genome. It won't package the spliced messenger RNAs. Okay, how about viruses that don't have a specific packaging signal? So picorna viruses, they don't have a specific sequence region in their genome that interacts with the capsid proteins. And the mechanism seems to be that the capsid protein, which one is it now? I've forgotten. Yeah, VP3, has an interaction with the 2C protein, which is part of the RNA replication complex. So the RNA polymerase is the 3D here, and all of it is kind of associated together with these other non-structural proteins associated with the endoplasmic reticulum. And this is the complex that is replicating the virus RNA. And these 14S capsid pentamers, they associate with the replication complex, and they start binding to the newly synthesized messenger RNA. That is helps seed the encapsulation and then the final maturation step to give an infectious virus particle. Okay. Also true that this is another Google Translate that I didn't pick up. So no. Okay, flaviviruses also don't have a specific packaging sequence. Okay, how about DNA viruses? Okay, so some DNA viruses have specific packaging signals. So that's in adenoviruses. There are packaging sequences at either end of the DNA genome. So the idea is, there's one at the beginning, one at the end. You try and get the whole genome in there. Polyomaviruses, not very selective. Okay, it seems like probably it's just selected by size. So you've got the capsid pentamers that are imported. They'll start to associate with chromatin, but on the cellular chromosome, you can't really get a closed structure. So you'll never get a particle. But with a small virus DNA genome, then you'll have enough capsid pentamers associated together to give you a close structure with DNA on the inside. So it seems to be just like selection by size. And with the herpes viruses, it's the terminase, which is connecting DNA replication to packaging. So the terminase will cleave off linear copies of the double-stranded DNA genome, and then it's going to associate with the portal complex and feed the DNA into it. So it's a protein-protein interaction, which is a specificity here. So the terminase complex has a specific interaction with the portal. Yeah. No, they don't. They don't have one. It's just coupling of replication to packaging, kind of like the picorna viruses. Okay, so the last example is for influenza virus, also mix of viruses. Now the problem they have is that in order to get a viable virus, you have to have one copy of each genome segment. And if you've got like something like this, where we've got 2 copies of segment 3, we don't have a copy of segment 1, this would be a defective particle. So about 20 years ago, there was still a bit of a debate about this. And there were some people who said, yeah, the virus is controlling the incorporation of the different segments. And each virus particle that's released from an infected cell, they've got one copy of each one, a whole genome. But there are other people who said, no, that's not true. It's random. And the reason it works is because, in fact, we don't really have 8 copies of each segment in a particle. We've maybe got 10 or 11. And it's a random selection of 10 or 11. And among these, you know, some of them will be defective, but you'll have enough of them. They've got at least one copy of each segment, so that will be okay. The virus will be able to replicate. So this was a discussion, you know, about 20 years ago or so. So one of the types of experiment that really ended this debate was looking very closely at the virus particles. So is it true that we have 8 segments per particle, or do we have 10 or 11? So let's have a look very closely with cryo-electron microscopy. So this was a paper published almost 20 years ago, and they looked at influenza virus particles just as they're being released from an infected cell. So the process is that the nucleocapsids will align underneath the plasma membrane, associate with the matrix proteins and the virus envelope glycoproteins, and then the particles will bud off from the surface of the infected cell. And if you can catch them while they're kind of like this, you get a particle that looks like that, okay? And also what you can do is, if you're very lucky, you can get ultra thin sections through these, through an individual virus particle. So you can look and you can see we've got 1, 2, 3, 4, 5, 6, 7, 8 genome segments in here. So each one of these is virus RNA together with the nuclear protein. And you don't ever find any with 10 or 11. You always have 8. And because the different segments are different sizes, you could say, oh, this one's segment 1, and this one's maybe segment 2, because this is the longest one, right? This one will be segment 3. So you can figure out that, in fact, it's got one copy of each. So this is telling us that orthomixoviruses, they do control which genome segments go into a virus particle. There's a way to ensure they have one copy of each one. And that's the way that they can have a full genome inside each virus particle. So how does this work? So fortunately, we've been able to figure this out. So what seems to be the case is that the ribonuclear protein, when you've got one copy of the negative stranded genome segment with the nuclear protein, it forms this kind of helical complex in a kind of like elongated stick form. But it seems like you've got bits of the genomic RNA that are kind of bulging out from time to time. So you have bits of secondary structure in the viral genomic RNA that are presenting bits of RNA stem loops, and the bases here can pair up with bases on another RNA segment. So what seems to be happening is that inside the virus particle, you've got these RNA molecules interacting with each other, okay? And that's what gives the specificity, that you have specific interactions between the different RNA segments. Okay, so that's kind of complex solution to a complex problem. Okay, so last part is release. How do viruses, once you've formed, once you've got the genome into the capsid, how is it released from the infected cell? So in non-envelope viruses, the kind of classical idea is that non-envelope viruses at the end of the replication cycle, they will destroy the host cell, okay? And the infected cell will be lysed. So that's true for adenoviruses. It's a lytic cycle, and at the end of the cycle, they express proteins that will destroy the host cell. So ADP is for adenovirus death protein, and it deconstructs or destroys the nuclear pore complex, and that's what will allow full capsids to be exported from the nucleus. So if you've got adenovirus infected cells here, they're accumulating whole virus particles like this inside the nucleus. They're too big, they can't get out, okay? They can't get out of the nuclear pore complex, but ADP is going to come here, destroy this, they can come out, okay? And L3 has an effect on the cytoskeleton, will destroy the cytoskeleton and allow these particles to be released from the lysed cell. So there's lytic infection with specific virus proteins that are destroying key components of the cell to allow release. So that's one way for non-envelope particles to be released. The other way is to have secretion. And this is true for poliovirus and other, maybe not all, but certainly poliovirus, one of the picornaviruses can do this. And they manage to be secreted by subverting the autophagocytosis pathway. So during autophagy, you have parts of the cytosol that are incorporated into an autophagosome. And normally what would happen is this autophagosome would fuse with the lysosome, right? And then everything that's inside gets destroyed. However, during poliovirus infection in the cells, the first steps occur, so you get the autophagosome, which is produced, but it's diverted to a secretory pathway. And so these virus particles that were initially within the cytoplasm here, they get incorporated into a kind of autophagosome, and then they're secreted. And so the particles are released. Now, how about envelope viruses? So they are all released by budding from a cell membrane. Now, the cell membrane can be the plasma membrane, or it can be the endoplasmic reticulum membrane, as we saw for dengue fever virus and the flaviviruses. It's also true that this is kind of annoying, that you can get, okay, I'm not going to talk about this because it's a bit too complicated, but herpes viruses have got a particular problem, right? So, yeah, I will talk about this after all. So herpes viruses, because we had herpes viruses as an example, they're producing their capsids in the nucleus. They put the DNA in there, you've got a mature capsid. So for herpes viruses, they're envelope viruses, they've got to get out of the cell. That's not easy, right? Because they're right in the nucleus. So the first step, now if you remember, the nucleus is a double membrane structure. So the first step is going to be budding across the internal nuclear membrane. And you get envelope particles in here, and then they fuse with the outer membrane. So you get kind of budding event here, and then these capsids delivered to the cytoplasm. And then you get association with the tegument proteins, and the whole thing is going to be associated with the glycoproteins here, and you get the mature particles with a second event of budding at the endoplasmic reticulum membrane. Yeah, I think that's true. Okay, so herpes viruses are a bit of a special case because they have two different cell compartments where the nuclear capsid has to be released by budding from a cell membrane. Most other envelope viruses just happens once, okay? So what are the different mechanisms? Well, first, you could imagine you've got a fully complete nuclear capsid with the genome inside, and it will acquire the envelope glycoproteins at some kind of membrane. So what is the process that's driving the budding is an interaction between the envelope glycoproteins and a fully formed nuclear capsid. So that's true for alpha viruses and also herpes viruses, okay, because it's a full capsid. So herpes viruses can be added here. Now, the other case is where it's the matrix proteins interacting with the virus genome, which are driving the budding. That's true for retroviruses. The third case is when it's the envelope glycoprotein just on its own. That's all you need. And that's true for hepatitis B virus. And then for also mixer viruses, influenza virus, then it's matrix proteins. That's these little blue circles here. They're essential. But you have other virus proteins that increase the efficiency. Okay, so I'm going to look in the last 10 minutes about in a bit more detail on HIV budding and release. So here's the process. Maybe I should have kept more time for this. You've got the full-length RNA genome copies, which are exported into the cytoplasm. With the packaging sequence, they can associate with the nucleocapsid protein here. Okay, so the virus RNA gets associated with GAG or GAG-POL polyproteins. Okay, and then the matrix protein directs these complexes to the internal face of the plasma membrane. So why does that happen? Well, the matrix protein has got N-terminal myrisdylation. So myristic acid, it's a kind of it's a fatty acid, so it's a post-translational modification that directs proteins to the internal face of the plasma membrane. So that's why you've got the matrix is essential here for that. Okay, so then what happens? Then you have the particle that's formed as you get more and more GAG and GAG-POL polyproteins that are added to this complex. You get the virus particle budding from the plasma membrane, and finally it's going to be released. Now, one of the things that people noticed when they compared the sequences of the GAG and the GAG-POL proteins was that in GAG you've got matrix, capsid, nucleocapsid, and then this weird thing called P6. Whereas in GAG-POL you've got matrix, capsid, nucleocapsid, protease, reverse transcriptase, integrase. And okay, matrix, okay, that's important for the budding. Capsid is to like produce a capsid. Nucleocapsid is to bind the RNA. We understand what the functions of all these things are. P6, what on earth is this thing doing? And it must be doing something because it's conserved. In all strains of HIV-1, even though it's a very variable virus, the P6 sequence is still conserved. So what happens if you mutate it and take it out? Well, what happens is that the budding process occurs, but these particles can't actually be released. You get a whole particle, it's formed there, but it's still attached to the cell membrane. Can't actually be freed up. And so you say, okay, so that's what P6 is doing. It's responsible for this very, very last step, the cleavage and release of these budded particles. So once you understand that, you can kind of mutate it in all different regions and see what different parts of it are doing. So in the P6 protein in HIV-1, there are two signals that are important. These like PTAP and LYPXL motifs that are present in HIV GAG-P6 and in other proteins involved in or essential for virus budding in HTLV1, the other human retrovirus, VP40, so in Ebola virus and VSV. All of these different envelope viruses have what are called L domains for like liberation of the virus particle. And their function is they're essential for the very, very last step. It's like nipping off of the bud here. So their function is to recruit cellular proteins in what are called the escort complex. So PTAP recruits TSG101, and these other motifs recruit other proteins, Alex and Ned, that form this escort complex. And the escort complex forms these proteins around the base of the budded virus particle, and then in the end, cuts it off. Okay, so it's the opposite of membrane fusion. Okay, membrane fusion is for entry, budding and release. You have to do the opposite of membrane fusion. And it's not virus proteins that do this, it's cellular proteins. So why do cells have these proteins? What's their normal function? Anybody got any guesses? Things that eukaryotes have, bacteria don't have. Vesicular traffic inside the cell. Okay, when you've got transport between the endoplasmic reticulum and the Golgi complex, you have these small vesicles, they're moving between them. And of course, these escort proteins, they're essential for the formation of a small vesicle that buds off from a larger structure in the cell. So all cells must have these types of proteins, and envelope viruses just, exploit them for this very last step in their release. Okay, so one other thing to say about release of envelope virus particles, it's not random from all the surfaces of the cell. So for influenza virus, you get preferential budding at the apical pole, not at the basal layer. So this is a virus infecting differentiated epithelial cells. And it's got transmission by people coughing, sneezing. It's got to be secreted preferentially back out onto the surface of the respiratory epithelium. So most of the particles that were released are from the apical side. And that will, yeah, okay. And for HIV-1, so something similar when you've got interactions between an infected T lymphocyte and an antigen-presenting cell, then you get preferential release at the contact site between these two cells. So virus transmission is very, very efficient here. It's not like you've got infected cells, they're just releasing a load of virus randomly into the blood or the extracellular fluid. I mean, that happens, but it's targeted, okay? So you get much more efficient transmission from one cell to the next. Okay, and very last step here is that sometimes you have a maturation of virus particles after their formation and release, and this is by proteolytic cleavage by the virus proteases. So for HIV-1, it's the protease that will cleave these GAG and GAG-POL polyproteins into the functional proteins. Adenoviruses have got something similar like that's something similar. So they have a protease that is incorporated into the capsids, and inside it will cleave virus proteins to give a mature infectious particle. Influenza virus, one thing that happens is that neuraminidase will remove the sialic acid from all the glycans on the glycoproteins on the surface of the virus. That's important because otherwise they're just going to stick to each other, right? The hemagglutinin will bind to sialic acid on glycoproteins, but it's also a glycoprotein itself. So you could have like two virus particles. If you don't have this activity, they're all going to stick to each other and stick to the surface of the infected cell. So the maturation of the virus particle to allow it to be released effectively involves, getting rid of the sialic acid on the surface of the virus particle. Okay, we're out of time, so I'll leave that question for the end. One last thing about this. Why is the protease activity, why does it have to be right at the end? Okay, let's think about this. So if I go back to here, imagine if we've got the protease that is active straight away. What's going to be the problem here? Probably we don't have time to understand what the problem is. Okay, to get to the plasma membrane, you've got to have the genome in RNA and you've got to have the matrix protein. But the genome is bound down here to the nucleocapsid. And it's the matrix protein that's connecting everything to the plasma membrane. So if you've got the proteolytic cleavage too early, like here and here, they get cut into individual proteins, the virus RNA is just going to be bound to the nucleocapsid. It's never going to be incorporated into the particle. So that's the reason why you've got to have the proteolytic cleavage right at the end. So I don't really know why the protease is inactive until the particle is released. And I think it might just be because of a kind of like a molecular proximity. The active form of the HIV protease is a dimer. So if you've got one GAG-POL polyprotein, just got one monomer of protease, it's not active. And in the cytoplasm, the chances of two of them like meeting and being close together is very, very low. But if you've got this kind of thing here, the particle is enclosed, you've got enough copies of GAG-POL for two protease subunits to meet up and start become active. So I think that's the way it works, really. But I'm not sure anybody has a really, really good mechanistic explanation. But that's my best guess on this one. So that's why it's important that the protease activity is later, okay? Because otherwise, the whole thing won't be able to form the particle. Okay, so time's up. I guess we see each other soon, next week again, I guess.